Okay, so today we are going to start on pedigrees, but before we do that, I wanted to go over a couple of concepts that I think people got turned around on from some of our genetic diseases. So the first one that we're going to review is Huntington's disease. So just a reminder, this is our only dominant disease that we were talking about. So this one is autosomal dominant, meaning it's not on the X or Y chromosome. It has nothing to do with whether you're male or female. And the thing that people were getting kind of crossed was that when I said someone has Huntington's disease, a lot of people were wanting to give that person two capital H's. And so for whatever reason, in the case of dominant diseases, you basically never see a homozygous dominant. They don't survive. So I just want to emphasize that whenever somebody says they have Huntington's disease, assume that they are the heterozygote, where they have those mixed alleles, and then start from there. So if I say that a father has Huntington's disease, you have to assume that it's the heterozygote. And so we'll give him one uppercase H and one lowercase h. And in this case, our disease alleles are flipped. Some people were getting mixed up because most of the time it is the two lowercase alleles that are causing the disease for our recessive ones. But for Huntington's disease, when you have little h, you are normal. So just a reminder how you set up your crosses for these: with the disease, heterozygous; without the disease, two lowercase alleles. And then you would set up your cross from there. So it was just that was throwing people off, that they didn't know which alleles to use when. The other question that people kind of routinely tripped over were the Punnett squares where the progeny die. So I think in the Go Formative it was something about creeper chickens. And the problem did say up front that chickens that were homozygous for the dominant allele did not survive. And for some reason, a lot of people set up crosses that were homozygous for the dominant allele. So if the problem is straight up saying that some homozygous is not surviving, then you cannot set up the cross with those homozygous mutants. So you would have to start with the heterozygote, right? And then there was another problem like this, that many people assumed the two uppercase alleles, but the parents could not survive. So those had to be heterozygous in your example on the homework, right? So be careful when you're setting those crosses up. Also, a lot of people forgot to cross out the babies that were homozygous dominant, and then they didn't notice that changed their alleles. So we'll go over that in class again. We'll look at that Go Formative. So just be careful with those, that if it says a homozygous whatever, it doesn't survive, then that means it cannot be a parent because it would not have survived to adulthood. And you also have to remember to cross those out on your Punnett scores. All right, on to pedigrees. So a pedigree is this sort of crazy diagram with all of these circles and squares on it. And what is actually looking at is this is following multiple generations, and it's looking at the inheritance of some disease. So humans can't, spawn by themselves. We need to have a friend. So males and females are represented on these graphs. Circles are always females, and squares are there to represent males. If an individual has a disease, they're going to have a dark color. If they do not have the phenotype of that disease, then they have the open color, the no color. And every line in pairs implies a pairing. So this would be mom and dad to these children here, and then this daughter would have married some other random dude and had a bunch of children. So that's how the overall architecture of a pedigree works. So we're going to be looking at how you distinguish between 4 different kinds of inheritance patterns in a pedigree. So we'll look at autosomal recessive, autosomal dominant, X-linked recessive, and X-linked dominant, because it is possible to figure out what sort of inheritance you're looking at just by looking at the pictures. So I'm going to give you some rules to sort of live by, and use these rules to fill out your chart. So an autosomal recessive, you will see an autosomal recessive when you see it skipping generations. So in this example, we have one affected parent, the trait disappears, and then it reappears in another generation. So autosomal recessives will always have this sort of skipping. And typically, autosomal recessive diseases, remember, autosomal means it's not on your X or Y chromosome, it's on one of the other 22. So it should be equal numbers of males and females. It might not always be, but generally speaking, you might have equal numbers of males and females. The other giveaway that you're looking at an autosomal recessive is that you will have two unaffected parents mysteriously producing an affected child. So if you see that every other generation the trait shows up and you have unaffected parents magically producing an affected child, that's probably an autosomal recessive. The other giveaway is sometimes the problem will tell you straight up that it's an autosomal recessive inheritance. So if it is sickle cell anemia or cystic fibrosis, then you automatically know that it's an autosomal recessive inheritance pattern because those diseases are both autosomal recessive. And when you see these pedigrees, the first thing that you should do is go about and start setting up the genotypes. So for an autosomal recessive, once you have established that it's an autosomal recessive, you can go through and for sure assign the genotypes of the affected individuals. If it's autosomal recessive, they have to have 2 copies of the lowercase disease allele, so you know that for sure. The other individuals that you can tell for sure are parents. If the parents have a sick child, but they don't have that disease, we know that from our Punnett squares, right? The only way to get that is if the parents are heterozygote. They would have had to have at least 1 copy of the uppercase allele to be healthy themselves, but they would have each had to have a lowercase copy to have a child with the disease. So from there, it is possible to go through and figure out the other genotypes. For this parent here, we don't actually know if they're heterozygous or uppercase, but because this parent here has 2 alleles, 2 lowercase alleles, this mom had no choice but to give each of her children one. She can either give this one or this one, so each of her children had to have inherited at least one of the little alleles. And because none of them are sick, they had to have an uppercase allele, which had to have come from dad because they don't have the disease, right? They don't have the 2 lowercase, so they had to get 1 lowercase from mom. And because they don't have the disease, it means they automatically have to have an uppercase allele from dad. And then the status of these children is not known, but you can calculate percentages for them. So that's the autosomal recessive. Skips generations, has unaffected parents having an affected child. You can assign immediately the people with the disease, those lowercase alleles, and you can usually work out from there at least some of the genotypes for the children. So an X-linked recessive is going to look a little different. And the big giveaway for an X-linked recessive is that you're going to have more males than females affected, because remember, males only have 1 X chromosome, so they're going to be more susceptible to these X-linked diseases. So in this pedigree, I'm going to count my males. Remember, males are the squares. So I've got 1, 2, 3, 4, 5 males who are sick, and only 1, 2, 3 females who have the disease. So this gives a strong indication that I'm dealing with an X-linked recessive. And then the other thing that you want to check to be sure that it's an X-linked recessive is you look for an affected mother, like this one here or this one here. If it's an X-linked recessive, a mother who is affected will have 100% affected sons, because remember, they get their X chromosome directly from their mom. They get the Y chromosome from the dad. So if it's really an X-linked recessive, every time you see an affected female, all of her sons, all the sons will have the disease. So if we look at this mom here, sure enough, her 2 boys, both of them have it. And when we look at this generation here, again, both sons did inherit it. So that's the other giveaway that this is an X-linked recessive. The other way that you will know it is an X-linked recessive pedigree is if they tell you, hey, this is a pedigree of red-green color blindness, which we already know to be X-linked recessive, or they'll say this is a pedigree of hemophilia, which again is X-linked recessive, so you already know the inheritance of those 2. So once you have established that you're looking at an X-linked recessive, the first thing you want to do is go through and label your males because they can either have the disease or they won't. So every affected male for an X-linked recessive, you know their genotype is going to be X little by. So you can go through and label every single affected male because they cannot have any other genotype but that for an X-linked recessive. And same thing for a healthy male. You can go through and label all the healthy males once you know that it's X-linked recessive. So this is definitely going to be an X uppercase BY, and so will all the other males that have that that are healthy. So from there you can go label the affected females, which will have to be homozygous for the disease allele. So you go through and you label all your females. And then from there you can kind of establish the genotype of the rest of them. So for example, this mom right here, her son is healthy. One son is healthy who has the normal allele, and one son has the disease. So both these boys could only get their X chromosome from mom because they got the Y from the dad. So that means that this mother has to be heterozygous. She can't be anything else because she has a son with one of each. So she is a mixed allele. And so we knew the genotype of this mom was homozygous for little b, and this was a healthy male, so we were able to label him as X capital BY. So when we look at this female, she was always going to get an X little b from mom. So she has no choice but to get one of those. It's the same allele from mom. And she only had one choice to get it from dad. So if she's healthy, she had to have the heterozygous allele. Actually, both daughters would have gotten the same because they can only get the X big B from dad and one of the X little b's from mom. So these had to be heterozygous. Can't be any other way. And so this female right here, let's see if we can figure that out. So she had to get X big B from her dad, but she could have gotten either one from mom, so we actually don't know for her. So you won't be able to solve all of them, but you can still calculate a possibility of whether she's heterozygous or homozygous. So that's the X-linked recessive. So you're looking first, did they tell you it's red-green color blindness or it's hemophilia? If not, did you count and then they had more males than females? And then once you get there, check the daughters that are affected or check the moms and look, do they have 100% affected sons? Because if they do, then that is an X-linked recessive. Okay, so the next one is autosomal dominant. So a giveaway for an autosomal dominant is that it's in every generation, which actually does look the same as this X-linked recessive. So it's going to show up in every generation. You might have equal numbers of males and females, but don't rely on that because it will come up sometimes that is not true. The way that you tell an autosomal dominant from basically the X-linked diseases is you look for the affected fathers. And then you look at their daughters. Because remember, the father has to give his X chromosome to the daughter. He doesn't have a choice. So if this was on the X chromosome, all the daughters would have the disease. But in this case, the father has some daughters that don't have it, at least one. And so this is saying that this is not on the X chromosome. And because it's showing in every generation, this is going to be an autosomal dominant. So that's the trick that you're looking for. So for autosomal dominant, look for the affected fathers, and do all the daughters have it? If they don't, this is an autosomal dominant. They might also straight up tell you it's Huntington's disease. And if they tell you it's Huntington's disease, you know it's autosomal dominant. It can't be anything else. And so once you have established that it's autosomal dominant, you go through and you label your healthy individuals first because you know they have to be homozygous recessive. If they had one copy of that disease allele, they would be sick. So go through and label all of the homozygous recessive. And now you also know that there's at least one disease allele for all the affected people. Again, they're probably going to be heterozygous. So that's how you solve the autosomal dominant. The autosomal dominant and the X-linked diseases can look a lot alike. And so if it is an X-linked dominant, it's also going to show up in every generation. But how you tell them apart is what is happening with those fathers. So because in an X-linked dominant, you might have the case where you see more females. Possible, but not always. But with an X-linked dominant, the fathers will always have an affected daughter, always. Because again, the dad will always pass his X chromosome to his daughters. So he didn't have any choice. So to this daughter, this dad gave an unaffected X, but she still got it because mom had it, right? So if I look at this affected father though, he had to give his X chromosome to his daughters, and both of them got the disease. And I know it's not mom because her sons are fine. So she looks like she had 2 good copies. If she had even one of these sons would probably have gotten it. So once you establish that it's an X-linked dominant, you know the unaffected individuals are going to have X little b if they're female, or X little by if they're male. So the easiest ones to label are the males. So diseased males will have X big BY. Unaffected males will have X little by. The females are going to be a little trickier because they could be mixed allele or double dominant. And the way that we tell that is basically based on what's happening with the daughter. So I know she has at least one dominant allele, but this son is healthy. He got this X chromosome directly from mom, so this mom had to be heterozygote. This daughter had to have gotten a little b from her dad. And because the other option for mom was a little b, she had to have gotten this X upper B from mom. This daughter's healthy, so she got 2 copies of the non-disease allele. So that is how you solve the X-linked dominant. And again, for X-linked dominant, you look at the affected fathers. If 100% of the daughters are affected, then that is an X-linked dominant. So those are the rules, right? Okay, and so get a picture of this. To solve the pedigree charts, you do need to have this information quickly available to you, so make sure that it ends up in your notes. Take a picture of it right now before we do the practice problems. Autosomal recessive. They'll tell you straight up it's sickle cell anemia or cystic fibrosis or some other disease that's autosomal recessive. The giveaways for this one are that it skips generations, it can have equal male and female affected numbers, and you will always find that heterozygous parent, or not always, but if you see an affected child and they have parents who are fine, that is another strong giveaway that you're looking at an autosomal recessive. The X-linked recessive, if they tell you it's red-green color blindness or hemophilia, we already know those inheritance patterns, but if they're asking you to solve it, you should look for more males to say that it's X-linked recessive. And if you see an affected mom, if all of her sons have it, then it's an X-linked recessive. Autosomal dominant, every generation is affected. But you can distinguish autosomal and X-linked dominant by looking at affected fathers. Affected fathers in an autosomal dominant will have some daughters that don't have the disease because it's not on that X chromosome, it's something else. But an X-linked dominant, you will. So for an X-linked dominant, the dad has no choice but to give that X chromosome to his daughters. So if you have an affected father and all the daughters have it, you're probably looking at an X-linked dominant. So as far as assigning the genotypes to the pedigrees, we did go over this in the video, but I'm just going to go through ahead and give you a list of steps. So once you get to these questions, first things first, did they tell you that it was a disease that you knew the inheritance to? Because you start there. And if they didn't tell you, you have to go through the little list of rules that I gave you to figure out which inheritance it actually is. And once you figure that out, then you kind of start following these rules. So if it's an autosomal recessive, So say they say it's sickle cell or cystic fibrosis. You go through and you label all the people who have the disease with the two little alleles, and you label all the heterozygotes that can produce that affected individual. If you decide that it's an X-linked recessive because they told you it was red-green color blindness or hemophilia, or you went through and found little rules and decided that it was X-linked recessive, label your males first because they are the easy ones. If they don't have the disease, they have X uppercase allele Y. If they do have it, it's X lowercase whatever Y. And then you can go through and label your affected females with 2 copies of the disease allele. And after that, it's usually you can just figure out who's what by following the Punnett squares. So for the autosomal dominant, again, if that's Huntington's disease, or you figure out that it's autosomal dominant, go through and label every single healthy individual with the 2 small alleles because they have to have that homozygous recessive genotype in order to not have the disease. So for autosomal dominant, immediately go through and label your healthy people with the lowercase alleles. And then the last is the X-linked dominant. So again, once you decide that that's what it is, label your males first. Those are the easiest ones to figure out. And then go through and label your unaffected females. The only way they would have the lowercase alleles is if they did not have the uppercase one at all. So that's how you assign those genotypes. You will see the blood type pedigrees, but those are pretty straightforward. I think we're good on that count. So the last thing that I wanted to talk about today were basically nuclear disasters. So genetic mutations can occur for lots of reasons. You can get them just from regular polymerase going through and making errors, but there are lots of environmental exposures that we can have that will increase the likelihood that mutations will occur. This is where all of our alleles come from, right? So those mutations are any change in the DNA sequence that's going to give me a change in the RNA that will eventually give me a change in my protein, which will alter my trait at the end of the day. And again, we call those different versions of the same genes alleles, and those alleles occur because of mutations. So sometimes one of the environmental factors that can occur is radiation or mutagenic chemicals. So both of these, radiation and mutagenic chemicals, will damage DNA structure and cause either single base changes or some major base changes to give you those different alleles. So radiation for sure increases the rate of mutation. If you go to the dentist and you need to have x-rays, you'll notice that they stick you in this like lead apron and book it out of the room. They don't want to be exposed to that. It's causing mutations in their DNA. So there's lots of radiation that can cause these mutations. So one of them is ultraviolet radiation from the sun. That's why we're supposed to wear sunscreen to protect us from skin cancer. But other types of radiation like x-rays or radioactive materials will also really damage DNA. And this is just sort of order in like their level of destruction. So ultraviolet rays are comparatively weak compared to x-rays versus radioactive radiation that will really harm you very quickly. So these are able to penetrate basically right through you and damage a lot of DNA along the way. So there are also chemicals like those found in cigarettes or mustard gas or nitrous acid. So if you expose your tissues to these, that's another way that you can be causing mutations in your own DNA. So that's why they tell you not to smoke. And these mutations might be all over your body or in only one part. So depending on the kind of exposure you had, if it was, say, to your lungs, you're accumulating mutations in your lungs if you're smoking. But if some of those mutations make it into your germ cells, so either the sperm or the egg, then it will be able to be inherited by the other generation. So some of the mutations, if they're somatic, that is, if they occur in any cell other than your germline, it's just going to affect you. But if it makes it into your sperm or egg cells, it will be able to, those mutations will transmit to basically forever, children and grandchildren. So there were a couple of unfortunate incidents that did occur where lots of people were exposed to really high levels of radiation. So one of them was the bombing of Hiroshima and Nagasaki. So this was when America dropped nuclear bombs on these cities. Um, so these were nuclear bombs. It exposed them to huge amounts of radiation. So people who didn't die in the initial explosion did suffer a lot of health problems from being exposed to that radiation. It caused tumors in the people. So this guy has tumors like all over his arms. And some of those mutations made it into their germline, into the sperm and the eggs, because it did damage DNA. And so this caused an increase in stillbirths and malformations in children for many years after those bombings occurred. So there was another incident in 1986 with the Chernobyl nuclear power plant. So this power plant had an explosion, and this released radioactive isotopes into the air following the explosion of a nuclear core. So this spewed huge amounts of radiation into that direct area. Radiation was detected as far away as Sweden. So in the 4 kilometers that were just downwind of this area, all of the trees died because it was basically like melting their cells. But the big issue that came from Chernobyl was actually thyroid cancer. So local cattle had damaged thyroids, and for some reason, a lot of the people that were in this area, over 6,000 cases were associated with thyroid cancer, were reported after exposure to Chernobyl after that disaster took place. But it has been, they have moved people out of that area, and wolves have taken off. Nature has kind of come back to the Chernobyl area. So that's something. But radiation was very harmful to people. So that's it for pedigrees and nuclear disasters.